Okay. So I was not around last week, so I haven't gotten a chance to submit the exams to be scored. I will do that this afternoon, and hopefully it won't take more than a day or so to get them back, so I will post everything as soon as I have it. The learning objectives are now up. I was not able to get them up before this morning, but they should be all set now. So let me know if you still can't get them. That has a list of learning objectives for this unit. And I'm trying to get my Wi-Fi to start working, which it doesn't want to. So, all right. For the time being, I'm going to just go from here then. All right, so we are going to start in on GI. GI is the last unit, and even though our final exam is not going to be cumulative, it is going to build heavily on everything that we've covered so far. The gastrointestinal unit is going to incorporate some smooth muscle information because of peristalsis. It's going to incorporate a lot of nerve information because of the enteric nervous system, and a lot of the cellular information because of all the different types of cell types that we're going to have, as well as signaling cascades and transporters that will be involved in the absorption of our nutrients. So, the first thing I want to do is mention that it is a very large economical issue in terms of healthcare. More than $40 billion a year is spent on GI-related medical issues. Okay, diarrheal diseases are like the third leading cause of health problems worldwide. So problems with your gastrointestinal tract are very diverse and can cause lifelong complications. If you think about the first organisms before we started getting to be very complex, what's the first thing they needed to be able to do? They need to be able to get energy. So before we had highly developed brains, the first thing we had to have was a highly developed gut. Okay? So we're going to cover, we'll have a whole unit that is basically just information about the gut. We'll cover a little bit of the liver and the gallbladder, but a lot of our content will be very heavily concentrated in lecture 21, which is the absorption and secretion that's happening in the intestinal system, mainly because you don't get any nutrient absorption until you get to the small intestine. Everything before the small intestine is really there for processing and preparing all of your compounds to be able to be absorbed. So, we have a few different ways of getting those compounds to be absorbed, but the food that you eat is not capable of getting into your body without mechanical processing and chemical processing. We eat proteins, we eat large starches, we eat complex triglycerides and highly complicated lipids. Your intestine can't absorb those. It absorbs monosaccharides, it absorbs amino acids and very small maybe tripeptides. It absorbs individual fatty acids and very small lipids. Okay, so in order to get to the point that you will actually be able to get the nutrients into your body, we need to have a series of chemical reactions in order to get it small enough so that it can utilize the transporters in that lining. So this is a cross section. I guess I should just quickly go back and introduce a couple things we're going to talk about when we get to peristalsis. So starting at the mouth, that's where your food comes in, you have a series of sphincters along your gastrointestinal tract that are really there just to make sure that everything stays separate and that the food moves at a rate at which your GI tract can handle processing it, maximize the ability to absorb the nutrients, and not get overflooded with too much at one time. Okay, you have several different sphincters. You have an upper esophageal sphincter, you have a lower esophageal sphincter. So this part here, your esophagus, has a sphincter on either end. The upper esophageal sphincter, that is what you open in order to determine when you're going to swallow something. Okay? The lower esophageal sphincter is there for what purpose, do you think? Why would you want to have a sphincter that basically shuts off the esophagus down at the base?
To stop stomach acid from coming back.
To stop regurgitation of stomach acid, okay? Now, if you do have acid reflux, you will get a series of problems in the lower part of the esophagus, which I will talk about in a little bit. But the two sphincters are really there to protect the esophagus. The esophagus is nothing but a conduit. It's a conduit to get your food from your mouth down to your stomach. It doesn't actually do any kind of mechanical disruption. All it does is propel it further downward. There's also your pyloric sphincter, which is on the antral side of your stomach, so the bottom side of your stomach. What do you think the function of that sphincter would be? I didn't hear that last part. Until
keep everything in the stomach to make sure that everything is dissolved before you pass it on.
yes, the pyloric sphincter is there to make sure not only that the stomach is able to serve as a reservoir, but it also is there to be effectively a size limitation. Anything that is bigger than about 2 millimeters is not going to be able to pass through that pyloric sphincter. The purpose of it is because the stomach is really the first point where you're going to get a massive amount of mechanical disruption and chemical disruption of the food that you've consumed. The first part of your mechanical disruption is really just your teeth, but the amount of chewing that you do is going to be insufficient no matter how much you chew your food in order to get it to the point where it can pass through that pyloric sphincter. After the pyloric sphincter, we also have an ileocecal sphincter, and then we have internal and external anal sphincters. So we'll talk about those on Thursday when we get to the peristalsis material, but all of these sphincters are there really to make sure that you have a controlled movement of food throughout the gastrointestinal tract. Okay, now let's get into a little bit about the breakdown of your duodenum. This is a figure, I think from the Boron book, that really walks through what happens if you look at all of the different levels inside of the duodenum. The serosa is really just the connective tissue that's on the outside of your small intestine. Every single part of your GI tract has the same basic overall layers. They just have slightly different functions and therefore slightly different cell types as you move through the body. But they're all going to have a connective tissue that is on the outside. Inside, you will have two layers. The muscularis externa really has two different layers of smooth muscle. You have an outer layer, which is longitudinal, and an inner layer, which is circular. So the difference between longitudinal and circular, longitudinal you can think of as a slinky. They're going to contract along the same plane as your GI tract. Okay, so they'll contract laterally. The circular is going to be the part that's going to contract and kind of push in, sort of like what you see with your sphincters. They're going to work together in order to propel food preferentially in one direction. It also can be used to churn food. Okay, so the food that is going through your gastrointestinal tract is not only going to be moved from mouth to anus, but it is also going to in effect be regurgitated in small localized contractions in order to help cause it to mechanically be disrupted a little bit better so that you can get a smaller breakdown of those food particles. After that, we have your submucosa and you have your mucosa. So this is your submucosa. It shows a lot of the blood vessels. It shows the entrance of your bile duct because this is the duodenum we're looking at. And then you have another layer of muscle that surrounds the lamina propria or the mucosa. The mucosa is going to be the focal point of our lecture 21, which is all of that information about absorption and secretion. Your submucosa is going to contain a lot of the glands that are going to basically hold on to the content that will be secreted into the lumen, but the mucosa are the cells that are in direct contact with the lumen. These are the ones that will reside along the villi, and each one of those cells is going to have microvilli to increase the overall surface area. They will be on this interior lining of that mucosal surface and in direct contact with the food that is passing through the gastrointestinal tract. If you looked at it in a cross-section, this is just looking at it, I believe this one's from Guyton and Hall. It's just looking at it from a different direction. Here's your serosa on the outside. You have the longitudinal muscle and you have the circular muscle moving in from the outside, and then you have your submucosal region and the mucosal muscle as well as the mucosal area where you'd find your villi and your microvilli and all of the enterocytes, enteroendocrine cells, goblet cells, stem cells that we're going to talk about in terms of overall function and how they all work together to make sure that you get the most out of the food that you eat. All right, so how are we going to make our food usable? The first part is your mechanical disruption. The food that is on your plate is in no way in a condition for your body to actually be able to utilize it. It has to be broken up into smaller pieces, which you will get by mastication with your teeth, and you will get it by churning inside of your stomach. All the contractions of your stomach will help to further mechanically break apart the contents of the food that you ate. Chemical digestion, these are your enzymes. We have a whole ton of enzymes that are going to be released that are going to act in different parts of the body. You have two that are released in saliva. Anybody know what they are? Amylase, which breaks down starches, and anybody know the other one? Lipase, which breaks down fats, okay? You don't get any protein digestion in the mouth, and technically, you don't get any fat digestion in the mouth. okay? You get starch digestion in the mouth with salivary amylase, but you do have salivary lipase. The difference is it doesn't get activated until it actually hits the acidic compartment inside the stomach. So you get lipase activity to digest those fats in the stomach, but the lipase is actually coming from the mouth. Amino acids, your peptide, your protein digestion, that is going to begin in the stomach with the release of pepsin. Well, with the release of pepsinogen and the activation of it into pepsin, which we'll get to a little bit later. But also when you get into the early part of the duodenum, you're going to get a series of carbohydrate, protein, and lipid enzymes in order to break down everything to make it readily available for absorption. So I made a chart to kind of summarize the location of these enzymes, the enzymes that are typically found there, and what the targets of each of those enzymes are. These are going to be discussed a lot throughout the course of the unit, so it's a good idea to have a good summary starting point. Salivary glands, we already mentioned you have amylase and you have lipase. That's going to break down your carbohydrates and your fats. So starches and triglycerides, which is the predominant form that we're going to consume each of these. The stomach is where you're going to have activation of your lipase, and you're going to have pepsin that will start breaking down your proteins. The pancreas. So when I said in the small intestine you're going to start getting digestion with a series of enzymes, you do, because they're all coming from the pancreas. You have two main components that the pancreas is going to provide: a whole bunch of enzymes and bicarbonate. Okay? The enzymes are really there to do the bulk of the heavy lifting in terms of chemical digestion. You have a pancreatic amylase to further break down carbohydrates. You have lipase and colipase. Lipase is going to break down your fats. Colipase doesn't actually have any enzymatic activity. Its job is to stabilize lipase. It's a cofactor. It is also secreted as procolipase, so it's just like most of these pancreatic enzymes, most of them, especially the ones that will digest proteins, are going to be released as zymogens. They're inactive precursors that have to be activated. Colipase is no different, despite the fact that it doesn't actually function to break down fats. It's released as procolipase and does have to be cleaved by trypsin in order to become activated. Phospholipase is going to break down your phospholipids. We have trypsin and chymotrypsin that are both going to be breaking down peptides. The only enzymes that are released as active enzymes are your amylase and your lipase. Okay? They are already activated. As soon as they're released, they're ready to go given the proper situation, right? Because lipase needs to have that acidic environment in order to become active to function, but they are considered active enzymes. They don't have to be enzymatically cleaved in order to function. All of your peptidases do have to be activated before they're going to function. They're all released as zymogens. You get pepsinogen, trypsinogen, chymotrypsinogen. They all need to be activated, okay? And the intestine, we have a couple of other enzymes that are going to be released specifically inside of the intestine. The pancreas is releasing all of these enzymes into the early part of the intestine. So the intestine, the duodenum, which is the first part of your small intestine, is basically the recipient of all the stuff from the pancreas. You're not actually getting any digestion happening inside the pancreas. The pancreas is just producing everything and then sending it to the intestine where its site of activity is located. Enterokinase is also called enteropeptidase. It's the same thing. No matter which text you read, you might see enterokinase, you might see enteropeptidase. The purpose of this particular enzyme is to activate trypsin. It specifically targets trypsinogen, cleaves it, and activates it. So anything subsequently that is activated by trypsin, cleaving that zymogen to get some kind of enzymatic activity, means you have to have enterokinase, aka enteropeptidase, first. Without that, you don't cleave trypsinogen to activate into trypsin. Once you activate trypsin, trypsin can auto-activate itself. So as long as you have one active trypsin molecule, it can activate more trypsin molecules. So it can have a positive feedback situation, but to get that first one, enterokinase needs to come into play. There are also disaccharidases and dipeptidases that are going to be present along the intestinal lining so that you can take any of those polymers or, well, I should just say, they're disaccharides or dipeptides. So 2 amino acids together, 2 monosaccharide sugars together, because you need individual amino acids and individual monosaccharides, individual sugar molecules, are what those transporters are going to use to be able to get inside of your body. And when I say inside your body, I mean from the lumen, across the cell, into the interstitial space, so it can be taken up by the bloodstream and go to where it needs to go. Anybody have any questions about these so far? Some of you might ask, you won't actually be tested on this, but I have had people ask in the past, how does enterokinase become activated? There's actually another enzyme called duodenase that activates enterokinase. So it's a whole series of cascades, but for all our purposes, enterokinase / enteropeptidase is sort of the initial starting point, okay? So that's what you should remember for all intents and purposes for this unit. But there are other enzymes that activate that one as well, in the interest of being completely accurate. Okay, so trypsin and chymotrypsin, they sound very similar in name, they have different functions. Trypsin specifically, based on the fact that you have a lot of these, you have a positive charge that is in the side chain, which means you have negative charge aspartate that resides inside the trypsin pocket. This is going to attract those lysine and arginine residues, right? Having that negative charge side chain for this aspartate or glutamate, the purpose of these is really there to attract those positively charged side chains, which means trypsin preferentially cleaves at lysine and arginine residues, okay? It's going to cleave there, which means that at all of the enzymes that it activates, it's specifically looking for these 2 residues, and it will only cleave anytime it finds that lysine and arginine, because that's all that can fit in here. Chymotrypsin will specifically cleave at your aromatic side chains. So your phenylalanine, your tyrosine, okay? These are things that are going to be cleaved specifically by chymotrypsin because you see a lot of neutral, small amino acid side chains, serines and glycines. They're really, really small, so it leaves a lot of room for that aromatic side chain, okay? So for trypsin, you have acidic side chains that line the pocket so that it can cleave at your basic residues, which are your arginine and lysine and arginine. And then for chymotrypsin, you have small neutral side chains that line that enzymatic pocket so that your larger aromatic carbons are able to fit inside there. Okay? Anybody have any questions about this so far? All right. So let's talk about some of the different types of cells. This is actually looking at the colon, but I use this because it gave a good visual of the different types of cells that we're going to find throughout the small intestine and the large intestine. The surface absorptive cell, that's your enterocyte. Your intestine is lined with enterocytes, and the main goal of these, the purpose of these, is for the absorption of nutrients or ions and water. So they're going to make up the bulk of the cells that reside along your intestinal wall. Goblet cells, these are going to be the second most abundant, and they're going to secrete mucus. And the further along in the GI tract you get, the more mucus is actually secreted. Okay, so what we're trying to do with mucus is to do what? What's the purpose of having a lot of mucus being secreted into the gastrointestinal tract? Protection. And what else? Buffer. And effectively lubrication, okay? The food that you eat is going to be relatively tough. Even though you're going to be secreting a lot of fluids, you might consume maybe 1.5 to 2.5 liters of fluid per day. The amount that your intestinal tract is actually going to be processing is somewhere around 8 to 9 liters of fluid a day, and a lot of that's because of all the stuff that your body is secreting into that lumen. Mucus is a big player, not only in the protection of the wall of your gastrointestinal tract, but it's also there to help provide lubrication so that everything can continue to move smoothly. your enteric endocrine cell, also called an enteroendocrine cell or EEC. These are your hormone secreting cells. At least that's how they've traditionally been referred to, because they are enteric, meaning in your gastrointestinal tract, and endocrine, meaning it's releasing a hormone. Turns out they also have other functions, and they do actually connect physically to nerves. So it's probably a little more accurate to refer to them as a neuroendocrine cell, because they have dual functions. Stem progenitor cells and undifferentiated crypt cells, these are going to reside at the base of the crypts, and they are there, the stem cells are there really to serve as a starting point to replace any of your enterocytes, your enteroendocrine cells, or goblet cells as you move up along the villus. Most of them are going to become your enterocytes because the life cycle of your enterocyte is relatively short-lived. It seems that there's some evidence that your enteroendocrine cells can actually live for much longer, and it might actually be due in part to the function that some of them will be able to have as neuro-type cells. All right, so our gastrointestinal peptides or modulators. We have endocrines, we have paracrines, and we have neurocrines. All of these are going to function in a slightly different way. Your endocrine function really is anything that's going to be added to get into the bloodstream to travel somewhere in the body to a target that is away from the location from where it was secreted. So all of your hormones are peptides, okay? They can be released into the blood and they can go and act on some other target. Some of those peptides are things that are going to act in a paracrine fashion. So they're going to be released and they're going to act on their neighbor, okay? So we're going to talk about somatostatin and histamine specifically when we get into, I can't remember what lecture number it is. I want to say it's 18, the gastric and pancreas lecture. We'll talk a lot about histamine and somatostatin and how they are released and really only act in that localized area. Endocrine cells can release paracrine functions. They can actually function locally as well as going into diffuse targets. The neurocrine specifically, some of them obviously are going to be peptides, some of them are not going to be peptides. There is also new evidence that some enteroendocrine cells actually release glutamate as their neurotransmitter. These are going to communicate with enteric nerves that can then communicate through the vagus to send information to the brain about the contents of the intestine and be able to allow for a more immediate response to the ingestion of nutrients. So this is a visual representation of everything that we just said. Endocrine on the top, which means your sensor cell is going to release a hormone, go into circulation to its target cell. You have neurocrine, which is the communication of target cells specifically from nerves. So in this case, we will have a sensory neuron, likely that would be communicating with a sensor, a gut sensor cell. So your enteroendocrine cell can communicate specifically with sensory neurons to allow for an effector response downstream. And then paracrine is the release of that hormone that can act on neighboring target cells. This is not a one and done situation. These are not isolated. They are actually all working together at the same time. 3 different methods of communication, all functioning together in concert to achieve an overall goal. So this figure on the right is really showing you have endocrine signaling. Oh, where's number 1? I only see number 2. Okay, so number 2 shows endocrine signaling. Number 3 shows paracrine signaling, and 4 also shows paracrine signaling. Crosstalk between enteroendocrine cells or communication between enterocytes and endocrine cells. And then you also have neuronal afferents that can communicate directly with those enteroendocrine cells. So this one is a figure that was from 2015. This has been a more recent area of investigation. And you're starting to see a lot more evidence showing up of direct communication of these enteroendocrine cells with the enteric nerves. All right, I have a series of hormones that I want to talk about, and this is showing them in the order in which they were discovered. despite the fact that we have a decent amount of understanding of a lot of these hormones because we've known about them for 100 years, there's still a lot of information that we are actively learning at this point because the gut has really been studied from a hormonal standpoint. It was sort of understood that hormones were released as a result of contact with food. And then we just studied what happened with those hormonal responses. So starting with secretin, that was first identified in 1902, followed by gastrin, cholecystokinin, glucose-dependent insulinotropic peptide, GLIP. Same thing as GIP. GIP you might have heard of as gastric inhibitory peptide. It's a little bit more apropos to call it glucose-dependent insulinotropic peptide because of its overall function. But they are the same thing. It's just one of these things where as scientists start to discover more accurate representations of what a function of a particular molecule is, sometime that necessitates a change in its name. Motilin was the most recently discovered of the ones that we're going to talk about, and it's going to be really important for the overall movement at the interdigestive period between your eating sessions. In order to consider something a hormone, specifically, there are 4 different criteria that you have to demonstrate conclusively to show that it has hormonal activity. So for those of you that have taken endocrinology, this is going to be quite familiar. So what's the first thing? You have to have some kind of event, physiological event, that happens in one part of the body that's going to affect the activity in another part of the body. That effect has to stick around after you've severed all nerve connections between those 2 parts of the body in order to prove that it is not strictly a nervous connection between point A and point B. If you cut all nervous connection, there still has to be that relationship, that cause and effect relationship. If you get rid of the source of that response, so whatever physiological event is received at point A that causes that activity in point B, if you take that organ out, that point A organ out, you should lose all response, which means that the chemical that communicates with point B must have been produced and released from that first point. And if you identify what that substance is, if you can isolate it, and then you can produce that response by injecting it into the bloodstream at a proper dose, you should be able to artificially create that response in the absence of the initial trigger. Okay? If all 4 of these work, you have a hormone. So that's how we sort of discovered the different types of chemicals that scientists were able to identify in the bloodstream and specifically understand how each of them was acting on different parts of the overall physiology of the body, was by making sure that we understood how this communication was occurring. So, as I said, gastric inhibitory polypeptide, same thing as GLIP. The top 2 I want to point out is looking at gastrin and CCK. Gastrin and CCK are very, very similar. And it turns out you don't need the whole massive molecule in order to get activity. You really only need to get gastrin and CCK activity at all. You need those last 4 amino acids, and that's it. These last 4 have to be on the carboxy terminus, and it's that terminal tetrapeptide. Now, I say carboxy terminus, okay? Because you see it ends in NH2. That is not the amino terminus. That is a phenylalanine amide. Okay? That's why I put a little picture on the right-hand side. So instead of having it C, double bond to the O, and then a hydroxyl group, this has replaced that hydroxyl group with an amide. That is what is required to get gastrin and CCK activity. It's the tryptophan, the methionine, the aspartate, the amide of phenylalanine. That tetrapeptide is all that you need to get activation of your gastrin and your CCK receptors. Pentagastrin is the last 5. It includes that glycine. It is a synthetic type of gastrin that can be used clinically. So, if both receptors are going to act in response to these last 4 amino acids on the carboxyl terminus, how is it that we get specific responses of CCK versus gastrin? It turns out that is mainly due to this guy. That tyrosine. In order to get CCK specific activity, you actually need that peptipeptide. You need those 7 amino acids. And that tyrosine has to have a sulfate group active. It has to be a sulfated tyrosine at that 7th position before you get to the end. So if you only have the 4, you're going to equally activate both gastrin and CCK receptors. If you have a sulfated tyrosine, methionine, glycine, and that carboxy terminus tetrapeptide, then you will get CCK specific activity. It will no longer interact with the gastrin receptor at the same degree. CCK and gastrin can both interact with each other's receptors to a relatively low degree. They just have a much stronger binding affinity to their specific receptors. But if you only took out, and that has to do with overall 3-dimensional shape after everything has been completely folded, but all you really need are these last 4 to get activity of both receptors. And if you have the last 7 and that tyrosine is sulfated, you will very specifically target the CCK receptor and much, much less so the gastrin receptor. How do we feel about that so far? Okay. I think I actually get into these in a little bit later. So I'm going to move on to discuss gastrin and its overall physiological effects. Gastrin's big job is to cause acid secretion. Hydrochloric acid is going to be released by your stomach into the lumen of your stomach for the breakdown of your food, for the activation of pepsinogen so that you can get some polypeptide breakdown happening in the stomach, and also to destroy any bacteria you might have consumed. All right? It's a very inhospitable environment. The pH of your stomach can drop down below the pH of 1. Okay? Average is about a pH of 2 when you have acid being secreted. So it's very inhospitable to any kind of microbes that might enter that environment. It's a way to protect you. So your acid secretion is going to have multiple overall functions. Histamine can also trigger acid secretion. It's just that gastrin is a lot more potent. It does a much, much better job. So if you have a little bit of gastrin, you get a ton of gastric HCl. It also has trophic activity, which is like cell growth activity. Because your parietal cells from your oxidic gland inside of your stomach is really responsible for releasing that hydrochloric acid, gastrin also will trigger growth of those cells. So if you have persistent amount of gastrin released in the body, inside your bloodstream, it can actually increase the number of these parietal cells that exist and reside in the stomach. Gastrin comes in 2 different forms. You have little gastrin and you have big gastrin. Little gastrin is 17 amino acids. It's released by the antral cells of the stomach, which is why if you surgically remove the antrum of the stomach, you will get atrophy of those parietal cells because you are taking away one of the main sources of that paracrine activity of gastrin to stimulate growth of the parietal mucosa. The big gastrin is 34 amino acids. It has the same kind of function, but it's released by duodenal cells. These duodenal cells are going to release that big gastrin, which tends to stick around in the blood longer than your little gastrin. Your little gastrin is released in a higher concentration, a higher amount. If you have a gastrin-secreting tumor, because they do exist, where you can actually have a tumor that results in excessive amounts of gastrin release, that will cause hyperplasia as well as hypertrophy. Does anybody know what the difference is between these 2 terms? Yes. Hypertrophy is growth of cell itself. Specifically on the top, we're looking at normal cells. Hypertrophy is the one in the middle. It means that your cell is getting bigger. Hyperplasia means that you are increasing reproduction of the cells. So both of them are going to result in an increase in overall organ size. They're just going to do it by slightly different ways. The cell itself getting larger, maybe not increasing the number of cells, but all those cells are getting bigger, is still going to cause the organ to get bigger. That's hypertrophy. Hyperplasia is instead of 10 of these cells, now you have 50 of these cells. You're increasing the rate of reproduction, so you're getting a lot more of them present in a smaller environment, and that's also going to help the organ to continue to grow. Okay, so difference between these two, both of which are going to be triggered if you have a gastrin-secreting tumor. It turns out that that gastrin-secreting tumor has a lovely name of Zollinger-Ellison syndrome. Of course, it's named after the people that identified it, because when you spend your entire life investigating something, most of your discoveries are in small increments. So when you make a big discovery, slap your name on it. Okay, so everybody now knows Zollinger-Ellison. Gastrinomas are tumors that are not within the normal function of the gastrointestinal tract. And because of that, they are also not responsive to the normal feedback mechanisms that stop them from producing gastrin. So what happens if you have a gastrinoma? You have excess amounts of gastrin being released into the blood, so you get hypergastrinemia. So hyper meaning a lot, gastra meaning a lot of gastrin, and then emia in the blood, right? So hypergastrinemia is a result of these uncontrolled tumors releasing excess amounts of gastrin into the blood. And what happens is you have an increase not only in parietal cell mass, because it stimulates hypertrophy and hyperplasia of that oxinic gland, but it also increases the amount of acid secretion because you're increasing the number and the size of the acid-secreting cells. What happens if you have that? You get a lot of acid in the stomach. If it's hyperacidic, you can get a peptic ulcer, mainly because of the overall function of that buffer lining that has bicarbonate and mucus in the stomach to protect that stomach lining. Too much acid is going to eventually be able to get through that point, but you're also going to have a situation where you have so much acid being produced in your stomach that as it starts to enter into the intestine, it should be able to be neutralized by the bicarb being released from the pancreas. If you have way more acid than your pancreas is capable of neutralizing, then you start to get problems of too much acid in the intestine, which can damage the intestinal wall because it's not prepared to handle that excessive amount of acid. So you'll get hyperacidity inside the intestine. One of the big problems of that is you're actually going to get something called steatorrhea. So diarrhea, everybody know what diarrhea is? Yeah, liquid stool, effectively, liquid stool. You have everything coming out of the colon is much softer, possibly even liquid, than it should be. Steatorrhea is basically fatty diarrhea. And the reason for that is because when you have a lot of acids coming into the intestine, when you get bile being added, and those bile salts, whose job it is to help to break down those fatty acids, they need to stay in solution. If they precipitate out of solution because of a very acidic activity, then they're not going to be able to be there to help to surround those fatty acids and prepare them to be able to be broken down by lipase. Lipase activity for pancreatic lipase. So remember how I said the salivary lipase needs that more acidic environment in order to become activated? Pancreatic lipase actually needs a more alkaline. It's optimized for activity in an alkaline environment because it's also being released alongside a whole bunch of bicarbonate. So if it needed an acidic environment for activity, that kind of defeats the purpose since they're both being released from the pancreas, and the pancreas produces a lot of bicarb. So pancreatic lipase needs a more alkaline pH in order to function fully. In this case, if you have hyperacidity in the intestine, pancreatic lipase, which really does a lot of work to break down your fats, is not going to be able to work as well. So you get less fat breakdown, your bile salts that are going to help aid in that fat breakdown have precipitated out. That contributes to less fats being digested and therefore absorbed. So you get a lot of fats ending up in the kind in the digestive that's actually getting removed from the body. Okay, so that's your steatorrhea. That is basically fatty diarrhea. So you get diarrhea because you have a lot of content that's not able to be broken down and absorbed. So more osmolytes means more water stays, which means more liquidy stool. Steatorrhea we just talked about, and hypokalemia. The other part about hypokalemia, which I actually think I have a question, so I'm not going to tell you yet. Am I correct? Yes, I am correct. Okay, I want you guys to brainstorm for just a second and talk to each other about why you think that somebody with Zollinger-Ellison syndrome might actually end up having a problem with hypokalemia. How could the changes in the GI tract as a result of Zollinger-Ellison cause a hypokalemic environment? All right. Very good. All right, I'm going to stop you guys. One of the big things about, I see a lot of decreased absorption, reduced absorption. That is, that's the gist of how we end up losing a lot of potassium. But why potassium? It turns out that your secretions, your gastrointestinal tract secretions, are actually very, very rich in potassium. So that's just a normal component of the secretions that are coming into the lumen that are supposed to be reabsorbed. If you still have the normal secretions going in that are very, very high in potassium, but now you are reducing the overall net ability of the gastrointestinal tract to reabsorb that potassium and to absorb its nutrients, you get a lot of potassium being lost through that watery stool. Okay, so low pH, as I said, this was just to sort of explain the steatorrhea side, and the hypokalemia specifically is because of a loss of specific GI secretions. It doesn't have to do with the potassium you eat, it has to do with the potassium that's already in your body that is naturally a part of the secretions that you are adding to your gastrointestinal tract. So if you normally are going to consume about the equivalent of 2 liters of fluid a day, remember I said there's like 8 to 9 liters of fluid that is actually passing through your gastrointestinal tract? The bulk of that's coming from you. It's being added and then it's being reabsorbed. Okay? So you're not actually losing a lot. You shouldn't be losing a lot. So when you have diarrhea, the biggest problem is that you're going to continue to lose a lot of ions, mainly potassium, as well as fluid, and become very quickly dehydrated because this is not supposed to be a main source of water loss. Okay. Fortunately, you can go to a doctor, sit in the office, and you can do a chirostim test, which is basically synthetic secretin. Secretin's job is to stimulate the release of bicarbonate from the pancreas, but it also has the function to inhibit gastrin release. It's part of the normal feedback mechanism that says we've already had digestion, we now have food that is entering into the small intestine. It's time for the pancreas to do its job, release those enzymes, and in this case, secretin will trigger it to release bicarbonate to neutralize that stomach acid. The feedback that happens normally in the body is to stop your endogenous cells from producing gastrin. Basically, stop acid secretion. If you have a gastrinoma, that feedback mechanism is not going to work. So if you inject somebody with chirostim, which is the trademarked or registered name of the actual compound, if you inject that into somebody with a suspected gastrinoma, if you still have an increase in serum concentration of gastrin above a particular level, then, or I guess to a degree above that baseline, that's going to tell you that you have a patient with Zollinger-Ellison syndrome. Because injecting secretin should stop the body from producing and releasing all gastrin. It should be that negative feedback that shuts down acid secretion by way of shutting down gastrin release. If you still have gastrin present and it actually seems to increase, then basically what happens is that gastrinoma is continuing to release gastrin. You have some other exogenous source of gastrin that is not subject to that feedback. Anybody have an idea of why you would actually see an increase instead of just keeping it neutral or keeping it at the same level? What's secretin doing normally? It's inhibiting the production of gastrin from the other parts of your body, right? So you're going to end up with less acid release. You're going to end up with a less acidic production of chyme and stuff entering into the intestine. Depending on where that gastrinoma is, it's basically that more alkaline chyme would indicate to that gastrinoma potentially that it could need more acid. So it might actually increase. It depends on where the location is of that gastrinoma. Sometimes it's in a location where it will sense the shutdown of endogenous gastrin, and it perceives it as an elevation of the pH and actually triggers an increase in gastrinoma release gastrin. So it'll either stay the same or it'll actually increase in the presence of this synthetic secretin. Normally, you should have it shut down. Okay. So that's gastrin. CCK. CCK's job is really to, it's the big trigger that says it's time to digest stuff. It's going to be sensed by enteroendocrine cells inside the duodenum that are receiving food from the stomach by way of the pyloric sphincter. It's going to sense your fatty acids, it's going to sense your proteins, and it's going to trigger the pancreas to release those exocrine enzymes. So that big enzyme push, all of those packages that are coming from the pancreas are triggered to be released by CCK. So it's going to trigger enzyme release. It's also going to slow down gastric emptying. It's going to trigger a tightening of that pyloric sphincter to slow down the amount of food that is coming out. Basically, it's sending the signal that the intestine now has a job to do, so wait your turn. So that you get a slow and steady release of food coming from the stomach into the intestine, which gives it enough time to be able to adequately digest and absorb. Emptying of the gallbladder. This is going to take the form of two different effects. CCK will relax the sphincter of Oddi and will also trigger contraction of the gallbladder itself. Okay, so if it's going to trigger contraction, most likely, which neurotransmitter would be released? Or which would you see an elevation of if you're going to stimulate any muscles to contract? Acetylcholine. What about relaxation of the sphincter of Oddi? Those are also muscles because it's a sphincter. Glycine would be one of your inhibitory neurotransmitters, but I'm thinking more globally. What is a particular molecule that doesn't need any kind of receptor or anything that we talked about in terms of relaxation of muscles? Nitric oxide. Nitric oxide is going to be the big player in helping to relax that sphincter of Oddi. It's also going to be a big role in helping your peristaltic receptive relaxation, which we'll get into on Thursday. Trophic effects, you're going to have an increase in the overall growth of the exocrine pancreas as well as the gallbladder. So CCK's big job is to get bile into the duodenum and get enzymes into the duodenum. So basically CCK senses food in the duodenum and causes the secretion and the release of a whole mess of stuff to help digest that food so that it is then in an appropriate configuration to be absorbed. So, fats, peptides, amino acids are coming into the intestine and they're going to stimulate CCK to be released. This is a big trigger for the duodenum because you really want to kind of get the ball rolling. This is where your pancreas is going to connect and your liver is going to connect. You have a common pancreatic bile duct in order to get this added into the duodenum. So this is triggered by food, and it's a chemical signal as well as CCK can also function electrically as a neurotransmitter, but I'll get to that in a second. to communicate with these two important organs that are necessary for overall digestion. That's why problems with pancreatitis or pancreatic cancer are particularly difficult to deal with, because it is so important for you to be able to have the materials produced by each of these in order to have proper digestion and absorption of materials that you are consuming. You can live without your gallbladder, because your gallbladder doesn't actually make your bile, it just concentrates it. It serves as a reservoir to highly concentrate that bile. So anybody who's had their gallbladder taken out, you're still fine, because the bile is actually produced by the liver. It's just stored in the gallbladder. But having it out does have other complications and does mean an adjustment to your overall diet. All right, secretin, as I said, this is going to be released by your S cells, and its big job is to inhibit gastric acid secretion. It's going to stimulate the secretion of your bicarbonate from the pancreas, as well as stimulating the release of pepsinogen. It does have a trophic effect on the exocrine pancreas, because the exocrine pancreas is the organ that its job is to release these contents into the lumen of the intestine. So when we get to the pancreas stuff, we're going to talk about, well, I'll introduce the difference between your endocrine and your exocrine pancreas. Anybody who did endocrinology, you probably talked ad nauseum about the endocrine pancreas. Okay, so the islet of Langerhans and all the different types of cells that are involved in the release of glucagon and insulin. We're not actually going to touch as much on that, because we're talking mostly about how the pancreas exocrine function for the release of enzymes and bicarbonate are helping with the digestion of your food. So, acid in the duodenum and fatty acids are going to stimulate secretin to be released, mainly because the big job is to stop acid production in the stomach and cause bicarbonate release from the pancreas. So basically, any acid that shows up inside the duodenum is a trigger for secretin to say, okay, we need to neutralize this, and we got to stop producing so much acid. So feedback signals, and because of that, it's considered nature's antacid. Glucose-dependent insulinotropic peptide. These are released from your K cells in your duodenum and the proximal jejunum. Fat has to be hydrolyzed, but basically all the foods are going to trigger this to be released. The big thing to note here, though, is that only oral glucose will trigger the release of GLIP. If you injected intravenous glucose, it's not going to trigger an increase in the release of GLIP, because the way the GLIP gets released is because it senses glucose in the lumen of the GI tract. So it has to come from inside of the lumen, not from inside of the blood. That's where the stimulus is going to be able to reach the cell, be sensed by the cell to trigger the release of GLIP. So that's going to trigger insulin release, and it's also going to inhibit gastric acid secretion. Motilin is released by your M cells that are also present in the duodenum and the proximal part of your jejunum. This is during fasting. So everything else we've talked about so far are hormones that are released in response to food showing up inside your GI tract. This is during a fasting stage. Motilin is really there to help for the interdigestive period motility. So remember how I said your pyloric sphincter doesn't allow anything more than like 2 millimeters to be able to get through it? That's during a fed state. How do you explain, if you're looking at something that is so small as nothing bigger than 2 millimeters getting through there, how do you explain somebody, well, hopefully not an adult, but let's say a kid who swallows a penny, how do you explain that actually being able to get through and out? You explain that by the migrating motility complexes and the movements during that interdigestive period, whose job it really is to clear out the lumen of the gastrointestinal tract. So anything that is too big for normal processing when you're processing food is able to be expelled from the body because of motilin triggering these different types of peristaltic contractions, considered housekeeping contractions as a way to just flush out anything that's left inside the body that's not able to be digested. All right, this chart, I want to say it comes from Guyton. It might come from Vernon Levy, I can't remember. But I wanted to highlight on the left-hand side, we have the stomach at the top that shows gastrin, and then, so this is the stomach, and then for this part we have the stuff that you see in the intestine. but I wanted to highlight specifically the different families, okay? So the gastrin family, these are a family of hormones. You have gastrin and CCK. So that should be abundantly clear considering how much similarity we've already talked about in terms of how they function and specifically how closely related they are and the fact that they can affect each other's overall activity. The secretin family is your GLP-1, your GIP, which is also GLIP, right? Your glucose-dependent insulinotropic peptide, polypeptide, and of course secretin. These are the family of secretin proteins. Secretin specifically, remember how we said we didn't need the whole molecule of gastrin or CCK? We only needed a small portion of it. Secretin, you actually do need the whole molecule, the whole polypeptide. It's like 27 amino acids and all of them are necessary. If you mutate any one of them, you don't get a functional secretin response. But this is really just a summary. It gives you the information about the stimulus for the release, the primary targets, the effects, and then some other additional information for each one of these hormones that we talked about. We didn't talk about GLP-1, mainly because that's released by your endocrine pancreas, and we're focusing more on hormones that are involved in exocrine pancreatic release. But we did talk about the top 5, so I thought it was good to include a nice summary chart. All right, motilin is your peptide family. This gives you some information here about distribution of GI hormones. You get antral release of gastrin, right? So you have those D cells in G cells in the antrum of your stomach, so that's the lower part of your stomach, right next to the pyloric sphincter. And you have gastrin being released in the duodenum. The rest of them are really going to be intestinal hormones, okay? The only one that's really released by cells that are present in the stomach of the ones that we've discussed so far is gastrin. All right. So that was our endocrine information. Now I want to talk a little bit about paracrine information. So somatostatin and histamine are both going to be big parts of the gastric information, the gastric and the pancreas lecture that we're going to cover. Both of them are going to work inside of the gastric mucosa, and their jobs are different in terms of what they're actually going to be causing. So somatostatin specifically is going to be stimulated by acid. The main reason for that is because it inhibits acid secretion. So when somatostatin cells, those D cells, are going to sense that there is an abundance of gastric acid, they will release somatostatin in order to slow down acid secretion. Okay, so this is the peptide that is shutting off acid secretion. Okay, so it's the acid-induced inhibition of acid secretion as well as gastrin release. Because gastrin triggers acid secretion, somatostatin is a two-pronged approach. It directly stops parietal cells from releasing acid. It also stops the antral G cells from releasing gastrin, which would then go and trigger the release of acid. Okay? So somatostatin's job is really to sense acid and stop it from being released or being triggered to be released, whether it's directly or indirectly. Histamine, as I said when we started talking about gastrin, also triggers acid release. It's not as potent as gastrin, but it does do a good job of triggering acid release. It's a paracrine fashion. The release of histamine locally will trigger your parietal cells to secrete acid. And I have a very lovely, somewhat complicated figure from the boron book that we'll get to when we actually cover what's going on in the stomach that will show you how all of these are kind of related to each other. So histamine is a way to trigger acid that's not as good as gastrin, but still does the job, which means that you can take a number of different drugs to stop an abundance of acid production in the stomach. You have a problem with acid reflux, maybe you want to reduce the overall amount of acid being produced. You can stop acid being produced directly. One of the ways that you can stop it indirectly is through a histamine H2 receptor blocker. So cimetidine is a good example. You do a histamine blocker, and that will stop that histamine triggered response for acid production. You can still have your gastrin trigger, and you still have your acetylcholine neurotransmitter signal, but you would be able to at least block the histamine, so it would reduce the overall amount of acid being produced. But both of these are working in paracrine fashion. They reside in the gastric mucosa, and they work locally on the gastric function of acid secretion. Neurocrines, so GRP is gastrin-releasing peptide, also called bombesin or related to bombesin. VIP and enkephalins, these are going to be located mostly in the gut. GRP is your gastrin-releasing, obviously is going to be in your gastric mucosa because that's mostly where gastrin is going to be released. These are all going to have different overall effects, but the purpose of looking at these is that they are neurocrines. These are released by nerves. So we've talked a lot about acetylcholine, we've talked about norepinephrine, but nerves have an abundant variability to be able to release different types of compounds. These are some of those neurocrine peptides that are released by nerves in order to cause an effect inside of your gastric or your intestinal mucosa. So this is not the most recent paper that came out. That came out about a month ago. But this was the first paper that indicated that there was evidence of enteroendocrine cells actually connecting to nerves, okay? And the main interesting part about this is that when we first started studying enteroendocrine cells, they were done by fluorescently tagging antibodies. And so the antibodies would just bind to parts of the cell. So all the drawings in all of the textbooks before then had this flask-like appearance. When transgenic models actually allowed us to illuminate an entire cell, the first thing that was noticed was this really weird sort of arm that sort of ran along the underside of the other enterocytes and seemed to have a physical connection with nerves. That connection has since been proven, and glutamate has been demonstrated to be the neurotransmitter that allows it to communicate, or at least one of them. So, but this was the initial indication that we do have enteroendocrine cells connecting with nerves. So depending on the text you read, you might read that enteroendocrine cells specifically are completely hormonal and that there's no connection. That is depending on when that was published, okay, and when it might have been updated. So the most up-to-date is that there's definitely a hormonal trigger, but there is also a neuronal component that seems to be related to how quickly the gut and the brain can communicate with each other. That takes me into the enteric nervous system. Your second brain actually has more neurons than the spinal cord, and it operates independently of the brain. If you sever all connections from the vagus, the enteroendocrine cell can still respond and trigger sensory and effector motor responses to the gut. The other thing is, it's like 90% of all the serotonin that your body produces. So when somebody takes SSRIs, you guys know what SSRIs are? Selective serotonin reuptake inhibitors. So antidepressants, it's supposed to prolong the amount of serotonin in the synapse to reduce the amount that's being taken back up to help trigger an increase in serotonin levels. The problem is it tends to come with a lot of gut side effects because the enteric nervous system actually produces most of the body's serotonin. And it's not specifically going to target just the nerves that produce serotonin in the brain. It's going to affect globally inside the entire body. So taking antidepressants do tend to come with a lot of complications, and one of those complications is issues with overall GI function, particularly in terms of peristalsis. We have an intrinsic and we have an extrinsic control. The extrinsic control we sort of already talked about a little bit. We have our sympathetic and parasympathetic control. Mostly parasympathetic is what we're going to talk about here because it's going to stimulate the gastrointestinal tract. Sympathetic is going to inhibit it, your fight or flight response. You don't really care about digestion of stuff going on in your gut, okay? The rest and digest, that's the one that we're going to cover mostly for autonomic control. The intrinsic control, though, we have the myenteric and the submucosal. Myenteric is also called Auerbach's plexus. It's primarily motor in function, and it resides in between the longitudinal and the circular muscle within the lining of the GI tract. Remember how we talked about you had that outer muscle layer, you had a longitudinal, and then you had a circular. The myenteric plexus is kind of in between there, also called Auerbach's. But I prefer myenteric, it's a little more accurate, and also it's easier to remember. Myenteric is primarily motor, submucosal is primarily sensory. Submucosal plexus, that's also called Meissner's plexus, and that is really going to hold on to predominantly sensory nerves to communicate any sensory information from the GI tract. This is not completely a sensory and motor divide. It's just that most of the information communicated by the submucosal is sensory, most of the information from the myenteric is going to be motor in function. So looking at it a little bit more closely, we have the myenteric plexus on the top here, and you can imagine we have the outer longitudinal on the top, and we'd have the circular muscle in between, and then you'd have the submucosal plexus that would be down below. Most of what we're going to have here is submucosals providing some sensory information and feedback, and the myenteric is going to be providing your more motor response. This one came from Nature Reviews. It's actually from Drug Discovery, so it has more to do with the paper actually has more to do with the drugs that are affecting these types of nerves, but I thought it was a good figure to show you the intrinsic peripheral afferent neurons, which is that IPAM, the red one. Shows you how that any information detected from the mucosal environment can be communicated to interneurons and sent to both the outer longitudinal muscle and then the inner circular muscle. So this is down here on the bottom, that would be like the mucosa, and the sensory information goes to interneurons, which allows it to communicate with both the outer longitudinal and the circular muscle to affect overall peristaltic contractions in response to food and stuff that is present inside of the GI tract. So it provides real-time neuronal fast communication between the status of what's inside your small intestine and how the muscle should respond in order to maybe promote better breakdown, so that we have maybe a lot and it's still kind of big. You get a lot of distension, that's a stretch receptor response, right? If you distend the intestine, there's a lot of food there, you activate stretch receptors, which communicate with these muscles in order to promote better churning, segmentation, and eventual propulsion. Okay, so this IPAM, I don't expect you to understand the specific response from this actual paper, except to understand that this intrinsic peripheral afferent neuron, that's what IPAM stands for, is communicating sensory information so that you can get contraction and receptive relaxation in order to promote the net movement and circulation of your muscles. All right, just a couple more quick slides. This just shows the blood vessels. It shows you the myenteric plexus here and the submucosal plexus. So the myenteric between your longitudinal and your circular, this is coming in closer to the muscle itself. The blood vessels are going to be in the submucosal plexus. This is just looking at it from a different arrangement to give you a more global perspective. The last part that I want to run through are the different types of salivary glands. So we will talk about these again, but you do have these facial nerves that we had talked about before, and the glossopharyngeal nerves, whose job it is to trigger submandibular, sublingual, and parotid glands to release saliva inside of the mouth in response to food or in response to the sight or smell or thought of food. This is not governed by any kind of direct sensory information from the tongue. It's governed by neuronal connections. So in the absence of actual food, you can have the release of saliva. Think Pavlov's dog. We only have a few more slides on here, but I can finish them up on Thursday. It's only 4 more. So I'll just finish those up at the beginning of class on Thursday. And if anybody has any questions, then please let me know. I'm sorry that I wasn't here last week, but I'm happy to answer any now.
No.